Hi, and welcome to OncoDaily TV. My name is Elen, and today we are going to explore the link between fruit flies and personalized medicine. It is my pleasure to welcome our guest today, Laura Towart, CEO of Vivan Therapeutics. Hi, Laura. Thanks for joining me today.
Hi, Ellen. Thanks so much for inviting me.
It is a pleasure. Without further ado, I want to jump into our topics today because we have very interesting things to discuss. You trained as a neuroscientist and later pivoted into oncology and precision medicine. What inspired that transition, and how did you arrive at the role of CEO of Vivan Therapeutics?
When I was doing my studies, I was at Weill Cornell Medical College in New York, and the program I was in was a tri-institutional program between Cornell, Rockefeller, and Memorial Sloan Kettering. And actually, even though my degree was neuroscience, my PhD lab was at Sloan Kettering, and it was in a lab that was studying molecular neuropharmacology. And my role in my PhD was really on kind of the molecular side, and so I got very interested in genomics. And of course, at the time, this is looking back now years, it was the advent of the genomics era, and everyone was, talking about the promise of personalized medicine. And I actually left my PhD to start a company called Cellmatics that was really a pioneer in personalized fertility treatments and guidance and treatment direction. Or maybe I'll just start that again. That's just that little part. So Cellmatics, we started off really looking at how to personalize and guide women to different fertility treatments. And so we did the first whole genome sequencing of a group of women who had idiopathic infertility. And we developed a test to be able to guide patients to one modality versus another. And so really from the start of my PhD, or by the end of the PhD, I was really interested in personalized medicine. And so I've continued that trajectory from women's health to then oncology. And really, I guess being in a lab at Sloan Kettering, I had a lot of interaction with patients, in the lobby and, throughout the building, and also with oncologists. And so at first I just had a general kind of compassion for the patients that were going through different therapies. And then I had a more personal connection later when, my grandmother and then my mother had colon cancer and unfortunately passed from colon cancer. That became more of a focus of my life, as I know I also have such a strong genetic component. And I realized that there has to be a better way of treating patients than what's currently available.
Oh, I'm very sorry about your mother and grandmother. You've been through a lot, and the way it turned out to affect your experience is very encouraging for others who are going through something like that. And you are very bold and successful to leave the PhD and create something completely very new, which has succeeded. So as a leader in a highly innovative field, what have been the biggest challenges and surprises you faced, both scientifically and operationally?
One of the biggest surprises, I think, early on with Cellmatics was we had a very, I'd say not easy, but it was relatively easy for us to raise our initial funding. But then as we were trying to raise more, we realized that there's such a big gap in interest in funding female founders, and then also a gap of people's interest in funding women's health. And I think so when I started Cellmatics, it was 2009. At that time, there was really not a big appetite for funding women's health. And now, of course, it's changed. And I think it's a really hot topic now. And there's more dollars or pounds going towards women's health, but still not as much as going, to other areas. And then I'd say with Vivan Therapeutics, I'd say still there's a very big discrepancy between investment in men and women and as founder. And so I feel like there's still a lot of work to be done there.
And that is one of the reasons, one of the many, you were recognized as one of 100 influential women by OncoDaily in 2025. And definitely your activity is inspiring more women and driving the change towards more funding and more focus on those areas. Who or what have been your greatest sources of inspiration or mentors along the way? And which lessons from them do you carry into your leadership today?
Oh, that's a good question. I'd say both of my parents were really like the main inspiration for, for everything I've done in my life. I'd say they were entrepreneurs, so they had their own business far away from the sciences, which they're probably smarter than I am, but just their perseverance, their dedication, their hard work, and those values that they've passed down to me, and also my desire to succeed, basically to try to make them proud as well, and carry on a legacy to show my children as well.
I guess that's the strongest inspiration coming from parents. Looking back over your career, is there a decision or turning point that you would say defined where you are today? I guess leaving the PhD was one of them, and the others.
There was another one. So actually, when I was in college, I was studying biology and English, but I thought that I wanted to be a neurologist. And so while I was doing my degree, I had to get some hours because I was going to be applying to medical school, and I had to get some hours in shadowing a neurosurgeon. And so I did that at George Washington University Hospital, where I was doing my undergraduate. And as an intern, you don't really get a lot of time with a neuro-oncologist. Oh, sorry. I need to start again. As an undergraduate, you don't really get a lot of time with a neurosurgeon because he's in surgery or she's in surgery. And so I ended up spending a lot of time shadowing the neurological physical therapist and interacting with the patients at that point. And so then I kind of changed my trajectory a bit to say, okay, I want to be a neurophysical therapist. And so I went to physical therapy school in Boston after that. And after a year in, I realized that it wasn't really for me. And because you don't just study neurophysical therapy, you have to study all aspects of it. And I felt like it wasn't scientific enough for me, actually. And so I got a job. Also I was in Boston. I had New York license plates, and I was getting a lot of tickets on my car. And there was a rivalry between the Yankees and the Red Sox. And so I feel like I was targeted a bit with my New York plates. And so I had so many parking tickets. My parents said, we're not paying anymore. You gotta get a job. And so I got a job at Harvard Medical School in a laboratory. And then I chose to go to Cornell. But that was also a big turning point. I don't know if it was the parking tickets. And then, so that really changed the course of my life.
You have a very interesting story, and we will get back to that in a moment. But for now, let's talk about Too Much, because it is as interesting as your personal story. What is it, and how does it aim to transform personalized cancer therapy for our viewers?
So Too Much is a tool that we've been developing since we've been laying the groundwork for Too Much since the inception of Vivan Therapeutics in 2018. And basically, the foundation of Too Much is all of the data that we've been generating over years by using what we call the personal discovery process. So the personal discovery process is our method in which we work with patients and also modeling tumors that we've seen from databases, where we look at a patient or whole exome or whole genome. We identify the genetic drivers that are driving that patient's unique tumor. We model them in fruit flies. So we create these personalized disease avatars, and then we expand the fly population to 500,000 or 1 million animals, and all containing, the same genetic makeup as the patient has for the tumor. And then after expanding the population, we take every drug that's ever been approved by the FDA, which there's about 2,000 of them, cancer and non-cancer drugs. And we also take investigational therapies. And we see which of these combinations are going to rescue the fly, because the fly is going to have a lethal tumor, and only the combinations that are the most effective are going to rescue the fly. And then we make a human treatment recommendation. And so the data that we've generated for each of these personalized disease campaigns, combined with all of the different therapies and the response, is a very large, unique proprietary data set. And so we've been expanding that data set so that we have enough coverage across colon cancer now, which is the focus of the company, and also gastrointestinal cancers. And so now we have the data set, and we've developed, we have world-class data scientists at Vivint, based actually in Armenia, where OncoDaily is, right? And we've now layer on top machine learning algorithms to be able to make recommendations that go beyond the data set. And so that product we call TwoMatch.
So this is like a mass clinical trial, but just for you. And
that's right.
And how do you validate and benchmark these recommendations against real patient outcomes?
So that's a good question. So TwoMatch is a tool that's using the data that we've been generating for other patients and over time. And so it's a software tool. So it enables us to go immediately from the patient's tumor sequence to a rapid recommendation. Whereas the personal discovery process can take time. It can take 6-7 months to be generating a unique animal and in doing such large scale drug screening. So TwoMatch enables us to really scale and offer this to many patients. And in terms of the validation, so the fruit fly is actually a very good model of cancer. So over 90% of the genetic alterations found in tumors have homologs in the fly. And so we're able to really model most of the pathways, the oncogenic pathways. And the fly is considered an animal, at least by the FDA. And so it's a very good, we get a very good picture of whatever drugs are working in the flies. We have a very strong belief that they're going to work also in the patient. And the fly is also a very good model of toxicity.
And why did you choose to start this in Armenia?
To be honest, I didn't really know very much about Armenia, and I still haven't had a chance to go, which I'd really love to. But we received some investment from Granatus Ventures, which is a local venture capital fund. It's a leading fund investing in tech companies around the world. And once they invested, they introduced us to the wide pool of candidates that you have in institutions in Armenia, like the Armenian Bioinformatics Institute. And so once we interviewed potential hires all around the world, and the highest level candidates that we found were actually coming from the Armenian Bioinformatics Institute. And so that's why we really started working there. Plus, it's a cost-effective place to recruit talent.
Well, I'm very happy to hear that as an Armenian, and I invite you to Armenia. We will host you and show you around. It's beautiful.
Thank you. And I've heard it's absolutely gorgeous, and I heard it's a great place to do remote work and visit. So I'd love to do that.
Looking forward to it. How does a patient purchase your services?
So up until now, patients have usually been referred through oncologists, but it's direct patient pay. So all of our services are offered only privately now, while we generate enough outcomes data to be able to apply for FDA and CE mark. And so once we have that, then we'll look to be integrated more. But patients now, we've started, because now that we have TwoMatch available, we've created a form on our website where patients can go, if they already have some type of tumor profiling, like a Caris or Foundation or Guardant or any of those, Tempus, if they have any report, they can upload it and they could actually see if we have a match available for them. So this is all patient can do on their own. And if we do have a match, soon they'll be able to click and download the report. And then that would come with a consultation. If a patient hasn't yet had any tumor profiling, we can coordinate that for them. We prefer to coordinate whole exome sequencing. But if the patient already has something that's pretty comprehensive, we can work with that. And we're also, patients can book a consultation directly on our website as well.
So from a clinical integration standpoint, how do you see TwoMatch being used in a real-world oncology workflows? How would you coordinate between the physician, the patients, the tumor boards, and Vivint's team?
So for TwoMatch, the report, we think it's very useful, and the oncologists that we're in discussion with find it useful. So it will have at the top of the report, it would say out of all of the standard of care therapies, it will rank them based on their efficacy in the avatar. And then, so we imagine that when a patient is first diagnosed, of course, the oncologist is going to have to do standard of care. And so we hope that our report will be able to direct them to the best of that, the most effective. Then we also test investigational drugs, and also drugs in the clinical stage. So there might be a trial happening somewhere, or there might be a single patient case study or multiple patient case study that's been reported, elsewhere. And so we list that kind of next. And so we rank those, and oncologists might have an easier time getting one of those combinations through their tumor board. Then lastly, at the bottom of the report, we have our unique combinations. And so we are able to identify usually unique combinations between cancer drugs and non-cancer drugs. And so it could be up to 4 drug combinations. And usually these are the ones that are the most effective. But we know that they also might be the most difficult for the oncologist to integrate into their treatment. So we provide guidance as to, the data that we've generated and how other oncologists that we're working with are able to access some of these combinations. So we've had success, for example, because the technology originated at Mount Sinai Medical Center, and they had a clinical study in which the novel combinations were used. The oncologists were able to get access via compassionate use using our report. And we've also had one of our patients so far being able to get access via compassionate use. So we're more than happy to help that process, and we really want the patients to be able to get access to those treatments. We envision that when a patient has started to fail standard of care, there's a much bigger appetite for moving to the novel combinations. And so we understand that now, and until we get more evidence for those combinations, it'll probably be a last resort.
And what is the difference between the personal discovery process and too much?
The personal discovery process is that personalized fly avatar development and personalized high-throughput drug screening of those avatars. So that's the process that takes 6 to 7 months. It could take sooner. So if we don't really know what we can offer a patient until we see their either exome or comprehensive profile. But basically, when we see that, we say, okay, this patient, let's say, has 6 genetic alterations. We can say, we've seen these 6 before, and we have a recommendation. If a patient comes and let's say they have 5 of the same alterations, but they have 1 new one, we can see quite easily if the the additional mutation is going to change the treatment recommendation. And in some cases, we can then say it's not going to be this, it will be that. But in some cases, we would say we're not certain if that additional mutation is going to change the recommended treatment regimen. So we would need to build a new animal. But in some cases, we have some of the genetic alterations already engineered in avatars. And so we don't have to recreate a whole new fly. We can, through genetic crossing of the flies, we can get the avatar that we desire. So in that case, it could take 3 months, not 7 months. And so once we see a patient's tumor report, we'll be able to guide them.
And what are the main scientific, regulatory, or business obstacles you see ahead of scaling to match globally?
I'd say the biggest obstacle for us has been funding. It's like always been funding. And so even we have a large European Innovation Council grant, and it's even difficult for us to access the full grant because we need funding alongside. And so it's been a real challenge. I'd say in terms of the regulatory process, we're able to offer too much commercially, and we offer the personal discovery process commercially. But of course, we want to go through the regulatory process so that we can access and make it available for more patients. We are currently generating the data sets to be able to do that. And we've had initial discussions with the FDA. We've changed part of the development of too much based on feedback from the FDA. And so we're working towards reapplying.
See, and if too much meets or exceeds your expectations, how do you envision its impact on cancer care over the next 5 to 10 years? What would success look like for you?
Well, success for me would be one that too much is really able to identify more effective and less toxic combinations and treatments for patients. And so really the combination of having an improved experience where the patients have better outcomes, and then they also have less side effects, and they're able to live happier, healthier lives while in treatment. That would really be like the most important for me. And also, right now we're offering 2-match for colon cancer, but I see this being expanded to other cancer types as well. So we've already started generating the data sets for 2-match lung and 2-match glioblastoma, and then maybe others after that. And so really I'm looking at success being able to treat most solid tumors and effectively.
And you recently announced about something new, imaging biometrics. Can you tell us a bit about that as well? What is the exclusive sneak peek information about upcoming announcements you have in the horizon?
Oh, sure. Yes. So we have a lot of exciting new announcements and developments. And so the most recent was the announcement we had with Imaging Biometrics. So they're a company in the United States that has gallium maltolate, which is their drug that they have initial evidence to show that it would improve outcomes in glioblastoma patients. And so I think it's just succeeded in its phase 1. But it's available to patients now through expanded access, I believe. And so we've gotten gallium maltolate now in our laboratory in London, and we're testing it against other tumor types. And so we have initial thought that this might be also useful in colon cancer and maybe other gastrointestinal cancers, and could be even others. And so because we have many patient drug screens ongoing, we can add gallium maltolate to the screens and see if it helps rescue the avatars. And then we can see which patient genetic signatures are the most responsive for population of future clinical studies for imaging biometrics. And then we also could potentially identify some combination partners that help work synergistically with gallium maltolate. So we're really excited about that. And it's really the start of a good relationship with Imaging Biometrics to work with them on their drug discovery and development going forward. And so that's very exciting. And we have a few more things that we're about to announce next week, I believe. We would just receive some grant funding from the Medical Research Council to work with something that we had a history of. So let me start that over. So the technology was developed at Mount Sinai Medical Center, and Professor Ross Kagan was the inventor of the technology, and he was also very interested in the potential of creating these personalized disease models with more comorbidities. So he basically created fruit fly avatars that had diabetes and also mimicked obesity, and engineered them with colon cancer. And so these fruit flies, the main output of this is that patients that have diabetes and cancer should likely get different treatments than patients without diabetes. And so that was published in Cell, I think in 2016. I'm not sure of the year, but that's always been an interest of mine, of how can we even make the flies more personalized and create more personalized recommendations for patients. And so this has been kind of brewing for some time, and we finally applied for funding, and we're now received it with. And actually the one of the students who was in Ross's lab, who was working on this, who was I think the first author on the Cell paper, he's now based in London, and he has a lab within the Medical Research Council, and we're collaborating with him on this. So it's a really nice kind of continuation of work that was done in the past. And we're now also offering this for patients. So if patients have diabetes and they get colon cancer, we can offer them the most personalized recommendations. So that's really exciting.
Well, congratulations on the grant and all of the success you've had. Thank you. I wish you good luck in the future announcements and future projects, because it looks like you have a lot in your plate, and we're going to definitely hear more of you. Asking something more personal, how do you find the power to do all of this, to drive the field forward? And what is the thing that drives you the most?
Oh, that's a good question. Sometimes I ask myself that. No, I think I really like my desire to make a difference is really driving me. And then also just, I really want to be a good role model for my children and just show them that they can, if they see something wrong in the world, or if they see something that they think they can improve, to go for it.
That's wonderful. And would you recommend 3 books to read for our viewers, or 3 movies to watch?
Okay.
Doesn't have to be cancer related.
Okay, 3 books or 3 movies. Let's see. Well, I could recommend one book. I have it right next to me. It's called Hacking Humanity, and it was written by Laura Lewington, and it just came out recently, and she actually discussed Vivint and our technology in the book. So for patients that are interested in hacking cancer. So I'd say that book, and trying to think what else. Maybe I'll have to go down the movie route. Let's see. Well, I really love the movie In the Mood for Love by Warkar Wong. I'm probably butchering the name. Yeah, that's one of my favorites.
That's perfect. I will make sure to watch that today. And I watched the book too. Thank you very much for joining me today. Thank you for the wonderful conversation. Who would you recommend to be interviewed by me next time?
I would recommend probably Ross Kagan, the inventor of our technology. He's now Regis Chair of Precision Medicine for the United Kingdom, and he's based in the University of Glasgow at the Beaton. And yeah, he's fascinating, and he's doing really incredible things.
All right. Thank you. I hope to see you soon and talk more about your very risky but successful career choices. And you seem to have a very, very powerful story behind it. Happy to talk about it next time.
Thanks so much. So nice to meet you.
Nice meeting you too.
Don't forget to like, comment, share, and subscribe to OncoDaily on YouTube. Hit the bell icon to stay updated.